All right, welcome Dag Sehlin to the Exploring Health and Medicine podcast.
Thank you very much.
Would you like to introduce yourself to the listeners?
Sure. My name is Dag Sehlin. I'm Associate Professor of Neurobiology, and I work as a researcher dealing mostly with Alzheimer's disease at Uppsala University.
Right. So I was thinking that we could go into, as an overview to start with, the clinical cognitive symptoms of Alzheimer's disease. Would you like to give us a brief overview of that?
Yes. So Alzheimer's disease is a quite common neurodegenerative disease, which is the main cause of dementia. And dementia is a set of symptoms that is common for several diseases, and Alzheimer's disease is the most common one. We also have frontotemporal dementia, Lewy body dementia, Parkinson's disease with dementia, etc. And Alzheimer's disease is characterized by memory loss and orientation deficits as early symptoms, and then later on problems with language, and also potentially at late stages with personality changes and so on. And there's also visuospatial symptoms involved. So it depends on which part of the brain is affected, and usually the temporal parts of the brain are affected first, and therefore the memory and the spatial abilities.
Right, including hippocampus, right?
Right.
Right.
So hippocampus is a very early site of neurodegeneration in this case.
Is that how doctors often first get the suspicion that this might be Alzheimer's through such temporal symptoms, so to speak?
Yes, I would say so. So normally patients are referred to a memory clinic from relatives who think that they have started to act differently, that they don't remember perhaps. Sometimes it's the person themselves that feel that something is different from previously, usually with regards to short-term memory. So they don't remember, where did I park my car, where did I put my keys, what happened yesterday. And sometimes also orientation can be the spark that gives the patient motivation to go to the doctor, that they come out from work and wonder, where am I? So that type of symptoms, and they are very much linked to the hippocampus.
Yeah, right. Can you give us a brief overview also of the progression of Alzheimer's disease throughout the brain? So it starts in the hippocampus, right? The progression from there.
Yeah, so it starts in the hippocampus, and then usually the rest of the temporal lobe is affected, and that's where the language comes in. And it's usually quite difficult for Alzheimer patients at a slightly more advanced stage to find words. That's also part of clinical assessment. And then later on it can spread both backwards to the occipital lobe and affect vision, or how visual cues can be related to memory and orientation. And then it also spreads to the frontal lobe. And in the end, some patients experience quite severe personality changes because of that.
Because of the frontal lobe effects?
Yes.
Yeah. Is this progress throughout the brain and the symptoms that are associated with that progress, is this how Alzheimer's is clinically discriminated from other neurodegenerative diseases?
Yes, I would say that these memory and orientation symptoms are the most discriminatory elements. But sometimes, or probably quite often, there are also comorbidities. So it can be quite difficult clinically to really distinguish Alzheimer's disease from a different neurodegenerative disease. Both because they can be similar in symptoms, but also because they can occur at the same time to various degrees. So if you ask a neuropathologist, they are usually of the opinion that pure Alzheimer's disease can actually be quite uncommon. There are usually some comorbidities, and then that also makes it more difficult with clinical assessment. And that's also why there is a big need for biomarkers where you can measure effects in either body fluids or with imaging and so on in the brain.
Of the Alzheimer's disease?
Exactly, and different diseases, so that you can be sure to separate them, which is important both to understand what is going to happen to the patient and also in order to potentially treat the disease.
Yes, right. What are some common comorbidities with Alzheimer's disease?
So, Alzheimer's disease patients can also have, for example, Parkinson-like or Lewy body-like pathology in the brain. They can also suffer from vascular changes. There is also vascular dementia, which is a quite common form of dementia. And then we also have frontotemporal dementia, and potentially also some other more rare variants. But they can, in principle, all mix into various degrees.
I see. So we've touched a bit on how Alzheimer's is diagnosed. You mentioned biomarkers in blood and in the cerebrospinal fluid. We also covered that cognitive function and different aspects of cognitive function, including language and so on, are affected at different stages of the disease. Are there any other parts of the typical, I guess there might not be a typical, or is there, way of finding an Alzheimer's diagnosis?
Well, I would still believe that the typical way of finding an Alzheimer's diagnosis is that a patient has started over time, it takes a long time, to develop a set of symptoms where memory deficit is perhaps the most prominent. They go to see the doctor. The doctor then makes a clinical assessment, and there are various tests that can be made. And the most simple one, the MMSE or MMT, is a questionnaire, basically where the patient is asked a number of questions which are related to short-term memory, to numbers, to words, and to remembering a sequence of words, for example. And this is a test that can be performed in the primary care. And then, if the test shows that this patient has a low score, then the patient can be referred to a memory clinic where specialists can do a more thorough neuropsychiatric assessment with perhaps a deeper cognitive set of tests. And then the patient can also have usually a lumbar puncture to assess biomarkers in the cerebrospinal fluid, or CSF, and potentially also a brain scan using CT or MRI.
Right.
So that, I would say, is the typical way of diagnosis of an Alzheimer patient. And with that set of samples, I think you can be relatively certain that there is an Alzheimer disease diagnosis that the patient should have.
Sure. And we went into the cognitive effects of the disease earlier. As regards the cerebrospinal fluid markers and the CT scan, what are the findings of the typical Alzheimer's patient?
Yes, so for cerebrospinal fluid, we have 3 markers that are commonly assessed. It's the amyloid-beta protein, which is what constitutes the main pathological hallmark of Alzheimer's disease. It's the amyloid plaques that are built up in the brain. And this is a process that starts long before the disease symptoms are visible. So amyloid-beta is a protein that aggregates, clumps together, and deposits in the brain as amyloid plaques. And while it does so, a certain isoform of amyloid-beta, which is called amyloid-beta 42, or A-beta 42, is lowered in the CSF. So because it is attracted to this plaque, mass of plaque in the brain, the levels decrease in the cerebrospinal fluid. This is a very specific sign of Alzheimer's disease. The other markers are tau and phosphorylated tau, or p-tau. And this is a protein that is associated with the brain cells or the neuronal cells and helps with transport of neurotransmitters in the neurons. And in Alzheimer's disease, this protein is hyperphosphorylated and aggregates into what is called neurofibrillary tangles in the brain. And when that happens, the levels of tau go up in the CSF and also phosphorylated tau. So A-beta 42 goes down, tau and phosphotau go up.
Right.
And with that pattern, you can be certain that this is Alzheimer's disease.
To my understanding, tau is increased during the later stages of the disease. Is that correct?
That's true. Or half true.
Okay.
There are different types of tau also, but it's true that amyloid beta comes first, and that is changed maybe 20 years before symptoms occur. And then tau comes a bit later, and it's increased, and increases also further on in the disease. So in that respect, it's true that it comes later. But recent research has also seen that certain fragments of, or certain phosphorylated tau fragments can occur also very early on, and also in blood. So that's potentially the future of Alzheimer diagnosis. That will
to just take a blood draw in the primary care unit and
yes,
make the diagnosis straight away from there.
Yes, at least to refer the patient to further examination.
Yeah.
I don't know exactly which function this type of blood biomarkers will have, but for sure it's a very interesting development, I would say.
Yes, I agree. Okay, so amyloid-beta 42 is reduced in the cerebrospinal fluid as a consequence of it being deposited into plaques.
Yes.
In the extracellular matrix of the brain. Is it correct that tau is increased, or at least hyperphosphorylated tau, in the cerebrospinal fluid as a result of tau going from intracellular location in the brain to the extracellular matrix and from there into the cerebrospinal fluid? Is that a useful mental model, or is it?
Yes, I think that's a useful model. And there is probably a link between the amyloid pathology and the tau pathology, which is also reflected in these early tau fragments that are released into CSF and also into blood. But it's still unclear what this link really is. So I would say that is one of the important questions in the mechanisms of Alzheimer's disease pathology that is studied. But it seems to be a causal relation between the amyloid-beta pathology and the tau pathology, which then releases these phosphorylated tau fragments to the extracellular environment and then to cerebrospinal fluid and blood.
So they're connected as well, but we don't really know exactly how.
Yes.
Yes, I see. You mentioned that tau in blood may be a future implementation of how we could detect Alzheimer's earlier. How soon is it possible to detect Alzheimer's currently? Like, can we detect it before symptoms currently, and is that the clinical case for some of the patients?
We can, is the short answer.
Okay.
By various means. And so the amyloid-beta pathology, for example, as we discussed, it can be detected in cerebrospinal fluid. It can also be detected with positron emission tomography imaging, PET. There are various PET ligands that can detect amyloid pathology quite early. And PET is a method that relies on a molecule that is radiolabeled with a minute amount of radioactivity. It's then injected into the body and it finds its target wherever it is. It's more difficult in the brain, but it's possible. And there it can then be detected with a PET camera that detects the radioactivity that comes from this PET ligand or PET tracer. And then with a computer you can reconstruct an image to find where and how much of this PET tracer you have in the brain, for example. And this technique has been very helpful in understanding the development of Alzheimer's disease in living patients over time. So there have been quite a number of investigations now over the past 2 decades where people have been examined longitudinally with this method, and it's been then possible to see how this pathology develops within the brain, like different regions, and also with time in relation to their cognitive status.
Right.
And with this we know that we could detect Alzheimer's disease long before symptoms occur. But this is a very expensive method, and you cannot just screen the general public in this way.
Yeah.
And that's where the blood biomarkers can be extremely important, I believe. Because with a blood biomarker, you can just take a blood sample, as you say, you can send it for analysis, and you can get an answer that is then relatively accurate. And with relatively accurate, I mean between 80 and 90% accuracy, probably. And this is not 100%, but
yeah.
But if you imagine that you would screen 1 million people and you want to find a few thousands of them for further analysis,
yeah.
or in this case quite many thousands, but still it's a cheap way to find the persons who need a more thorough diagnosis, I think.
Yes, yes.
So in the future, where there is a reliable treatment option and where you can treat probably before you get symptoms, I believe that this can be a very important way to find the patients early.
Yeah, I agree. That sounds hopeful.
Yeah.
How long before, like just a number or a range for the number of years before development of symptoms, can we find patients currently, and how long before clinical symptoms would that blood tests be able to detect or Alzheimer's?
Yeah, I think it can be a little bit difficult to say. So far, what we know about these things is from research studies, big research studies with many patients included. And there you can see that on average, you can probably detect PET amyloid pathology, for example, 10 to 15 years before symptom onset. But on the individual level, if you have one patient, I think it will be much more difficult to say, to give a number, but I believe that probably at least a few years before symptom onset, it could be possible. It also depends, of course, how often do you test. So do you test the patient or the public every 5th year, or every year, every 2nd year? And that is, of course, a matter of cost and benefit to the general public.
Sure.
I believe that if we would have such a program where you get tested, for example, from the age of 50, then maybe you would test at 50, and 55 and 60, and then perhaps with a narrower range the older you get.
Because the probability of getting the disease is greater the older you get.
It exponentially increases with age.
Okay.
So at the age of 80 you have now, I don't remember the numbers exactly, but maybe 20% risk of being.
Yeah, I see. So a major benefit of earlier detection of Alzheimer's would be the possibility of going in with earlier treatment, right?
Yes.
And recently, well, a little less than a year ago, Lecanemab was approved in the US by the FDA, and quite recently it was also approved in the in Europe by the European Medical Agency for treatment in early Alzheimer's disease. And you have played from my perspective, a central role in the development of this new drug. So congratulations on the success. Yeah.
Yes, this is I mean, obviously, for our research group, it's a big joy, and we are very happy about it. But I would say also for the research community, especially in Alzheimer's disease, it's also a very important step forward, because it's the first, and now there are also a couple of other antibodies, but this one is the first disease-modifying treatment against Alzheimer's disease that reached the market, and that has actually come to patient use. And I think that's very important, although the effects, the clinical effects on the cognitive decline of patients is not huge. That must be admitted. So it's not a magic cure, but it's the first step into disease-modifying treatments, and I think that's very important and quite big. So yeah, we're very happy about that.
Yeah. How effective is it in halting the disease?
Yeah, this is the main question, I would say, and a very important question. And here we also come to this question of how early can you detect the disease, and which patients do you treat. So I can give a little background history. So this antibody is one in a long line of antibodies that have been tested for treatment of Alzheimer's disease. And the idea is that you develop an antibody that should come into the brain and bind to these plaques, amyloid plaques, and eliminate them. And this concept of immunotherapy, which utilizes the body's immune system to clear amyloid plaques, it was invented for Alzheimer's disease just before the year 2000. So there was a publication in Nature where researchers had used a vaccine instead of antibodies, but with the same idea to clear amyloid plaques. When this was tested in patients, it gave severe side effects. So instead, it was concluded that it's better to develop antibodies that target amyloid-beta and then give them to patients in doses that can be determined directly. So then many antibodies were developed, and we also developed an antibody starting somewhere around 2003 or 4. But our idea was slightly different from the others, because we wanted to target the aggregated species of amyloid-beta, which are pathological. This is a protein that is constitutively produced in all people, and it probably has a physiological function, although we don't know exactly what it is.
Are we talking about the oligomer form?
Yes, so the oligomer form, or the protofibrils as we call them, slightly larger, they are toxic. So that's what we wanted to target.
Yeah.
So after some years of development, we discovered this antibody in the lab, and then we assessed it with preclinical studies in our lab, and then it was clinically evaluated, so tested in populations of Alzheimer patients. First small groups, and then larger and larger.
Yes.
And this was done by a company called BioArctic in Stockholm, together with a big pharma company, Eisai, in Japan.
Mm-hmm.
And these trials, as I said, they have been ongoing for 20 years almost. And in the beginning there was a lot of trials that failed.
Yeah.
So there was quite a lot of disappointment and in the field for a long time. But one reason why they failed is that patients weren't included in the right way. So in the beginning, they tried to treat patients with severe disease, which of course you can say that's probably where you could also see the most benefit.
Yeah, through some reasoning.
Yeah, exactly.
perspectives, Yeah.
And they are also the easiest ones to identify, probably. But it turned out that there were no effects in those trials. In early trials, there was also a difficulty in selecting the right patients. This is before amyloid PET was used to select patients. So it was found later that up to 30% of included patients in some trials did not have the amyloid plaque pathology in the brain.
Mm-hmm.
So if you treat someone that doesn't have your disease, you will not see an effect, obviously.
Yeah.
So,
but can I just
sure.
stop to make sure I also got the other point you were trying to make correctly, that I understood that correctly? It was not only that some of the patients didn't have amyloid-beta, but it was also that the patients that did have amyloid-beta had a severely progressed form of Alzheimer's disease, and the mechanism of lecanemab and other antibody-based treatments for Alzheimer's is slowing down of the building up of plaques.
Yes, and not only that, but also the effects that come after. So as we discussed in the beginning, amyloid-beta pathology has a link to tau pathology, and then also to neuroinflammation, which we haven't discussed yet, which probably also has a big effect on the whole neurodegeneration process.
Yeah.
And if you halt the amyloid pathology before it has started this cascade of other events, you are likely to stop them as well. Because it's believed that it's not primarily the amyloid-beta pathology that causes neurodegeneration.
Yeah.
Perhaps, or probably, the tau pathology is more important for that. So if you remove the trigger of the tau pathology, meaning the amyloid-beta pathology, early, then you're likely to prevent the other ones. But if they have already started and you take away the trigger, the other processes can go on anyway.
Yeah.
So that's probably at least one reason why it is important to start treatment early. And that is also something that was done with the Lecanemab studies, that they went for the earliest possible symptomatic Alzheimer patients, so mild Alzheimer. And that is determined with different types of cognitive tests. So the combination of starting early, having reliable inclusion criteria with amyloid PET, and targeting only aggregated forms of amyloid beta, but not the physiological monomers, are probably like the combination of factors that made this antibody successful in the end.
Lecanemab.
Yes. But interestingly, now, because you initially asked about treating early and how important is that, and if you look at the clinical phase 3 trial that was published with lecanemab 2 years ago, you can see that if you take a subset of patients, those that had the lowest amyloid and tau pathology when the treatment started, they benefited more from the treatment than those, or than the average of all patients. So that shows, or it indicates, because there are too few to statistically say this for sure, but it indicates that if you start earlier, you will not only have a better cognitive function when the trial starts, you will also deteriorate less over the time that the treatment is ongoing. So So there seems to really be a benefit of treating early.
Yes. I understand. And so you mentioned cognitive function. Are there any other clinical benefits? I understand that it's a disease-modifying drug, so it acts in the brain to reduce oligomers primarily, but also plaques, right? And you mentioned cognitive function, but are there any other clinical, like daily functioning, hospitalization rates, quality of life?
Absolutely. Those are very important things that you bring up, and normally, I mean, in the course of Alzheimer's disease, you will get not only the deficit in cognitive functions that we discussed, but also, as you say, reduced quality of life, the activities of daily living will be severely affected, and when you enter into the more advanced stage of the disease, you will need constant care, basically, or very much assistance, and that is also one of the big costs that are associated with this disease to society. And it's not only a cost in money, but also in time and efforts from relatives or next to the patient. So this is very, very important. When it comes to these therapy studies, I think that still they have been ongoing such a relatively short time, so it's perhaps difficult to draw much conclusions, or I haven't read so much about it, at least, with respect to quality of life and activities of daily living, because they start treatment at an early stage. So it normally takes a few years before you reach these stages of the disease where you really have problems with this. Having said this, I'm certain that for some patients it may have had a big impact, because it's important to understand when we talk about these effects, it's on a group level. So there are patients who haven't deteriorated at all after starting the treatment.
Yeah.
And then, of course, you don't know whether they would have without the treatment.
Sure.
But there are cases where patients believe that they have really benefited from the treatment, and it has kept them completely normal.
Sure, but there are placebo-controlled trials for this,
yes, there are. And it's and that's also why you need such a large number, because the variation will be very big between those that feel that they are normal, and maybe they would be also without the treatment.
Yeah.
But then, since you have also the placebo control where they don't get the medication, you can see that on a group level there is a difference.
Right. And so, as regards hospitalization rates and quality of life and daily functioning, in these large randomized controlled trials that have been carried out on lecanemab specifically, what are the effects?
Here I must admit that I don't really know these things, because these trials are carried out in multiple centers throughout the whole world, and although you can sometimes read about these things, it's not very often included in the scientific publications. They will be probably, but probably after some more time, when there is more data, because these things are not part of the primary outcomes of these trials, which are first of all cognitive functions, and then the biomarkers of different kinds.
Yes.
So,
How effective is lecanemab for those primary outcomes? Like, are we talking a halting of the progression with 50% or 80% or 30% or 10% or?
well, when it comes to clinical progression, the phase 3 clinical trial stated that it slows the progression by 27% cognitively at 18 months after treatment start. If you look at biomarkers instead, amyloid-beta, most of the patients are regarded as amyloid negative after the treatment of 18 months, which means that you have basically removed the pathology from the brain. So in that sense, it is very effective.
Yeah.
But as we discussed before, if you haven't started early enough, these other pathological events may still be ongoing and continue to deteriorate the brain.
I see. Could it be then that if, for example, lecanemab would be studied for 10 years, then the clinical rate of slowing the disease progression might be larger because the trigger has been removed, so there is no longer a trigger for the cascading neurodegenerative processes.
Exactly. And that's actually also what has been seen. Because the first trials, there was a phase 2 trial, slightly smaller, but people that were part of that had then been offered to continue. And also in the phase 3 study, those that were treated have continued on treatment.
Yeah.
So some patients are now up to 4 years, I think, in treatment, or maybe even more. And exactly as you say, you see that the difference increases with time. So there is a benefit of long-term treatment. And just recently, lecanemab was granted approval for continuous treatment in the US, which means that after this, the trial was only done for 18 months.
Yeah.
So the initial approval was also to treat for 18 months.
Yeah.
But then there is a time after that as well. And now it has been granted that you can continue. And then the patients are given slightly lower doses or less often.
Yes, yes.
So that is certainly something that has been seen, and that is, I think, quite logical.
Yeah, I agree. I know that. Well, I read that there was a survey that 85% of the surveyed Alzheimer's patients wanted I think it was the European Medical Agency to approve lecanemab. So this was before the approval. But I've also heard some, I think primarily some physicians that are not super excited about lecanemab, thinking that 27% as an effect size might be a bit small to risk the side effects that we will go into more soon. But it sounds then like the long-term effects might be larger than 27%, and that there is also mechanistic logic behind why such long-term benefits seem to exist.
Yeah, but I think, it's very sound to be skeptic, and I think that you should definitely listen to people who raise these concerns. 27% is quite a subtle effect.
Yeah.
And if you treat someone who falls below the 27%, because obviously this is the average, then it's even less.
Yeah.
And then of course you have some that really benefit. But the concern is definitely valid, I think, also with a high price, because it costs a lot of money. $26,000 per year is the list price.
Yes.
And if you treat many patients, it will take money away from treating someone else with a different condition. There is not an endless amount of money, unfortunately.
Yeah.
So I think the concerns are valid and reasonable. And then also, as you said, there are side effects and there are risks of side effects. And there are patient groups with higher risk. And then there is also like an ethical concern. Should we avoid to treat those that have a higher risk, which are also quite often the ones that are in higher risk of getting the disease in the beginning? And there the European and the UK medical agencies have come to the conclusion that you should actually remove some patients from treatment.
And now we're talking about the APOE4 double allele.
Exactly.
Yeah.
So people who have APOE4 epsilon 4 alleles, if you have 2 of those alleles, you're at higher risk of getting these side effects, and you also have a higher risk to get Alzheimer's disease in the beginning. So I think that is of course a concern, and it's sad for those people who have these, that you're in such a desperate need, and then you are not elected for the treatment. And I think it should also, as you said, 85% of Alzheimer patients were in favor of an approval, and I think that is an important part of this whole development of therapies, that there is such a desperate need, and it has given hope to both patients and relatives. So even if the effects are small, there is at least something now. And when you have something, you can improve it, and so on. So I think that's also an important aspect of this development.
Yes, I agree. And I didn't know that part about the growing benefits over time as well, like the larger effect sizes. At what time points were those? Like you mentioned, 4 years.
I believe that 4 years was one, but I'm not exactly sure about it. These findings have, as far as I know, not been published in scientific peer-reviewed papers yet. It's more communication from the company that has been given at conferences, for example, where they give out some data as it comes. And it's also, I believe, it's been the basis for the approval of continuous treatment.
Right, makes sense.
Which makes sense.
Yes. So we mentioned that there are side effects of lecanemab.
Yes.
Would you like to give us a brief overview of those?
Yes. So the most common and important side effect is something that is called amyloid-related imaging abnormalities, ARIA.
Yes.
And there are 2 types. One that is H, hemorrhage, and the other one is E for edema.
Yes.
So that means that when you get the antibody treatment, you can either develop small bleedings in the brain or small edemas where you basically have an influx of liquid into the brain. And you can see these as changes in MRI imaging. So for treatment of patients with these antibody therapies, not only lecanemab but also others, including, for example, donanemab, which is a different antibody, they require that you do MRI imaging so that you can see these changes. Mostly they are not harmful and sometimes completely asymptomatic. So it doesn't necessarily mean that something is super wrong and
it sounds scary though.
It sounds scary,
yeah.
And for sure it can be, and it can also be very severe. But usually it goes away if you stop the treatment. And then sometimes you can start treating again, but maybe with a lower dose, and then you can increase the dose gradually. And probably what happens is that the antibody accumulates in vascular amyloid deposits. So the amyloid-beta protein can aggregate in the brain parenchyma extracellularly and form plaques.
Yes.
But it can also aggregate in brain vessels, blood vessels. And it's not exactly clear why this happens, but it could be, or it's been hypothesized that it accumulates there because of a decreased degradation or clearance of amyloid beta from the vascular or perivascular spaces just outside the vascular wall. So antibodies that are administered, they have to be given at very high doses because otherwise they don't pass the blood-brain barrier, which is the barrier between the blood and the brain, which is there to protect the brain from pathogens and so on. So therefore the antibody has to be given in high doses, and that means that there is a risk that they accumulate in high concentrations where you have these amyloid deposits in the vessels.
Yes.
And when that happens, that you get accumulation in the vessels, there is recruitment of immune cells, macrophages and T cells and microglia maybe, depending on where you are. And this can cause an inflammatory reaction, which can even, to some extent, disrupt the blood vessel. And that can cause either a bleeding, or you have changes in the vessel walls that causes the edema. And in very severe cases, you can have almost like a stroke-like reaction, which can even be lethal. There have been a few deaths. It is maybe not to 100% certified that they are caused by antibody treatment, but I believe that it's quite likely. These patients had also comorbidities with vascular changes and in some cases also medication that is now regulated. But still, it can have potential terrible consequences, although extremely rare.
Right, extremely rare, and when compared to the potential benefits, and when you compare the data and the statistics of, okay, this many people will benefit this much, and you compare it to the side effects, I mean, the reason why it has been approved must be, I assume, I hope, and I believe, because the agencies have looked at the evidence and seen that the benefits outweigh the risks.
And I've heard, I'm not sure about this, but I've heard that it is perhaps not uncommon that the US medical agency and European medical agency have slightly different views on risk-benefit. So you can imagine that in the US you're more inclined to say that if the patient wants the treatment, we give it. And the patient has to decide themselves whether the risk benefit is good enough. I'm hypothesizing a little bit here, but I think it could be something like that. But here, if we go back to those that are excluded from treatment, the ApoE4, epsilon 4 carriers or homozygotes, the reason they are excluded is because they have a much higher risk of developing this vascular amyloid, and also therefore of developing these side effects that are caused by antibody treatment.
I see. Yes. So they have high risk for these ARIA side effects, and for that reason, it's not because it's less effective for these patients, it's rather that the side effects are...
I think the decision was made based on the side effects, but there are also indications that they may even benefit less from the treatment.
I see. Yeah.
Although I'm not certain that that this is completely confirmed.
I see. What are some other side effects of lecanemab?
I don't think there's a lot of other side effects that are specifically related to lecanemab. So I believe that other side effects are more related to treatment in general. I mean, you can get, for example, skin irritation, or you can get headaches.
Because it's infused into the...
It's infused, yes.
Yeah.
You get an intravenous infusion, which is also quite a big thing to do every second week. So it's a difficult treatment in that sense too.
Right.
But I don't think there are any other groups of severe side effects that can be linked to the antibody itself.
So it's the cerebral hemorrhages, the edema, and then it's mainly the cost of the treatment. Other than that hinders. Well, it is those side effects, but it's also the costs that hinders the patients that don't get the treatment from getting the treatment.
Yes, I would say so. And regarding the cost, we still don't know for sure when or if even it will be available here in Sweden or in Europe. And there I think the cost can be one deciding factor. What it will cost, how, will it be subsidized, and so on. And there's also a negotiation between each country's medical agency and the company that produces the medication about the cost.
Yeah, because it's gonna have to be well economically sound for the company as well.
Yeah.
I understand that. So these side effects, I assume, are dose dependent? Yes. And so one improvement, one iteration of lecanemab to reduce these, one iteration of these, one improvement upon lecanemab is to reduce the side effects through reducing the dosage, right? How are you trying to accomplish this?
We would like the antibody to get into the brain more efficiently. So this is, if we can achieve that, if we can give, then we can give a lower dose and still get more antibody into the brain.
Right.
And the key here is to pass the blood-brain barrier.
Yes.
So that has been our mission in our research group for the past 10 years, maybe, to develop antibodies that can penetrate the blood-brain barrier. And the method that we are using is called the molecular Trojan horse. So it means that we engineer the antibodies. So antibodies can be recombinantly engineered. Basically, you do it with a computer program.
Okay.
And then you can transfer this into a plasmid, a bacterial plasmid that carries the genetic code to build an antibody that you have designed in the computer. It sounds a little bit science fiction, but it's very well established techniques that have grown over the past decades to become very much available to researchers.
Mm-hmm.
So you basically design your antibody, you order a little plasmid that you can insert into a cell culture, and then the cells produce your antibody, and you can purify your antibody and characterize it and use it. So we have engineered the antibodies to carry a small antibody fragment, and this fragment binds to an endogenous receptor that sits on the blood endothelial cells in the capillaries of the whole brain vasculature. This receptor is called the Transferrin receptor, and normally it is involved in transport of iron into the brain. So the brain has a large use of iron for all its processes, and iron is toxic in its pure form, so it has to be carried by a protein.
Okay.
Transferrin. So that happens throughout it's carried in the blood by transferrin, and in order to get into the brain then, this large protein needs a specific carrier.
Transferrin,
yeah. So there's a transferrin receptor whose task is to transport iron on transferrin through the blood-brain barrier. So it captures transferrin, it transports it through the endothelial cell into the brain parenchyma, where the iron is released and can be used by brain cells. What we do is that we have an antibody that binds to a little domain on this receptor that is not used by transferrin. So we don't change the normal function of the receptor. We just hitchhike when it's going into the brain anyway. And then the antibody comes in and is released on the other side, and there it can bind to amyloid-beta.
Smart.
So, yeah.
Yeah.
So, and we haven't invented this ourselves. It's a technique that has been used or developed over the past decades. It was invented even in the 80s actually.
Mm-hmm.
But it's in the past years that it has really exploded also within the pharma industry to try to develop this for therapy. And there's one such antibody. Lecanemab is not yet in clinical trials with this type of brain shuttle.
Yes.
But there is one antibody from Roche that has reached clinical phase 2.
Mm-hmm.
And it actually looks like you can give a lower dose and more efficiently remove amyloid plaques from the brain of patients.
And I assume then also without so many side effects.
Yes.
Yes.
The number of patients is still so small, so it's too early to say that for certain that it is like that.
Yeah.
But all the data so far point in that direction. So
right.
That is very exciting, I think.
Yes. And that's the hope, and I believe that there are many more antibodies than just the one that you mentioned that has reached phase 2 that are also using this mechanism to
yes
get past the blood-brain barrier.
So I think basically that all medical companies that have antibody therapies for the brain, they also have a program for this, and they also co-collaborate in a way that is not always very easy to understand for people outside, but I think that over the past 10 to 15 years there has been a very much activities in this field of drug delivery to the brain, which was actually neglected for a long time. I think that when these developments of these antibodies started around 25 years ago, people didn't really think about this. They just, well, if we give enough doses, it will come into the brain.
Yeah.
But I think this type of brain shuttle mechanism will probably be necessary to make it safe and effective and available to a larger population. So that's an exciting development, and the work that we do, I mean, it's difficult to compete with the big pharma companies, so you have to have your little niche. So what we do is that we study how this affects the brain distribution of Lecanemab, for example, and also we are trying to develop this into a diagnostic method where we use an antibody labeled with a radioactive isotope to try to visualize its distribution in the brain with PET imaging.
Yes, and that will depend on how, or rather where it binds to amyloid-beta, right?
Yes.
Yes.
And as I said before, there are already very good amyloid PET tracers. So you might wonder, why would you like to develop another one? But the thing is that the amyloid PET ligands or PET tracers and the antibodies, they don't target really the same thing. So if you want to know, for example, that your patient really has something in the brain that your antibody will recognize,
yes,
it would be more accurate to detect it with the antibody instead of with an amyloid ligand that binds to something else, but within the same, how shall I say, deposit. So it's not completely different, but it's slightly different. But we're also interested in like developing this technique, and amyloid-beta is a good model system. So,
I see.
So perhaps we will not improve the diagnosis in the future, but we are developing sort of a new concept for brain imaging, because antibodies can target different things than classical small molecule drugs.
Yes, so not only amyloid-beta, but also other target molecules that might be relevant for other, for example, neurodegenerative diseases.
Exactly.
Yeah.
And one such thing is neuroinflammation, for example, as we touched upon a little bit, that neuroinflammation is a process that is associated with Alzheimer's disease, but also with basically all other neurodegenerative diseases. And it's a condition that is difficult to diagnose because there are no very specific biomarkers for it. So if we could develop a PET ligand that can accurately measure where and to what extent you have neuroinflammation, that could be very helpful for a number of conditions.
Sure.
And it could also, you could imagine that with different markers you can find different types of neuroinflammation, because inflammation is often discussed as one thing, but it has many different components, which can be disease specific or general to many diseases.
Yeah.
That's,
could you go into that a bit more in depth as regards the specific inflammatory processes in Alzheimer's disease? How are immune cells relevant for the pathogenesis of Alzheimer's?
Yeah, so in Alzheimer's disease and also many other diseases, but The brain immune cells, microglia, are reactive. So they react to the buildup of plaque pathology, and they try to counteract it by enclosing the plaques, but also to try to phagocytose them, to eat them, basically. We also have another glial cell, astrocytes, which is very abundant in the brain and which is also very important for helping neurons to communicate with each other. So astrocytes are involved in synapse formation, for example. They are also lining the whole vascular system. So they have end feet that are sensing and helping to cover the brain vasculature.
Yes.
So astrocytes are very important for a number of functions, and they also seem to be involved in both eating A-beta and perhaps also secreting it, thereby assisting in spreading. So these two cells are maybe the main players in neuroinflammation in Alzheimer's disease. But we also have peripheral immune cells, those that circulate in the blood, for example, T cells and macrophages. And they can also contribute to Alzheimer's pathology. It's still, so T cells, for example, have been found inside the brain. And it's a little bit unclear maybe how they get there, because they also need to penetrate the barrier. But there are ways for peripheral immune cells, such as T cells and macrophages, to affect the brain health, both from within the brain, but also from outside the brain. So they also accumulate around the brain, in the meninges and in the vasculature, and they can signal in a way that it's probably reaching the brain as well. This is, I would say, a relatively new area. Obviously it's been studied for a long time, but I think it's particularly hot right now, and there are new findings every week about how the immune system is involved in Alzheimer's disease and other diseases. But there's also genetic evidence for example, microglia and how they are involved in Alzheimer's disease. So there are mutations in a specific protein that is called TREM2, that is a relatively, which is quite much associated to specifically Alzheimer's disease. And this protein is a surface protein on microglial cells, which has been shown to interact with amyloid-beta oligomers. So in that sense, there seems to be a link between A-beta and TREM2. And TREM2 mutations by themselves can lead to Alzheimer's disease because of a dysfunction in this communication.
By themselves?
Yes.
So this is a familial, hereditary at least, form of?
Yes, and I don't know if it is completely penetrant. There are also familial versions of Alzheimer's disease that affect amyloid-beta pathology, and they are 100% penetrant. I'm actually not sure about the TREM2 mutations, but for sure there is a genetic component linked to microglia, which is Alzheimer's specific.
Right, and so when this TREM2 mutation exists in a patient on the surface of their microglia, what does that entail? You mentioned that entails phagocytosis, did I understand that correctly, of the amyloid beta into the microglia, and then what happens after that?
Yeah, this is a good question, and here I'm a little bit off my comfort zone, but I think that because I don't remember exactly what the mutation does, but it changes the way at least microglia help in preventing the buildup of the disease. Because neuroinflammation it's both beneficial and bad, probably. So initially, when you have an inflammatory response to an injury, it's beneficial, it helps to heal. But it's when it goes on for a long time and becomes chronic, with the release of different cytokines, for example, that can be harmful for neurons, for example, that it becomes a bad thing. So initially the inflammatory response is probably helping to degrade the plaque pathology, for example. And there is probably where the microglia that don't work as intended with the TREM2 mutation fail to contribute.
I see. Could you say that both for microglia and for astrocytes, and for neuroinflammation in general, it is a good thing to prevent or slow the early stages of Alzheimer's, but when these processes go awry, the progression is let loose? And
I believe so.
Yeah.
But this is also to some extent controversial. There are people also who say that maybe actually it's the neuroinflammation that starts the disease, and that amyloid pathology is a bystander or a consequence. And I think it's important to be open also to that hypothesis, because there could be, and it could also be that Alzheimer's disease is quite heterogeneous and that it can start with inflammation or amyloid pathology. So I think it's probably important to try to be open-minded and follow research progression and see what comes out.
Yeah.
But as it is now, I would say that the amyloid cascade hypothesis, as it's called, is the main hypothesis, where the amyloid-beta pathology starts. It gives rise to tau pathology and neuroinflammation, which probably, it could also be that the neuroinflammation is actually the link between A-beta and tau pathology. Because the glial cells are very abundant and important in the brain, and they as I touched upon, are involved in many different processes in the brain, in the healthy brain as well.
Yes.
So when that doesn't function anymore, you can imagine that it causes a lot of problems.
Yes. Do lecanemab or other antibody-based therapies for Alzheimer's disease that have been tested clinically, like in clinical trials already, or that are in more preclinical research stages, do any of these have any proven mechanistic effects on any of these neuroinflammatory processes?
That is a very good question, and also one thing that we try to study, because it has been suggested that when you treat with an antibody, so antibodies are part of the immune system, and their function is to find and identify an intruder, for example, and mark it for immune cells to come and eat it, basically, or eliminate it. And so there is a potential risk of this treatment and which is also potentially part of the reactions that we see with ARIA that we discussed, the side effects. But it could potentially also happen inside the brain. So if the antibody comes and it binds to a plaque and microglia come and eat it, because microglia are the brain's phagocytosing cells. Whether that then causes a release of cytokines, for example, pro-inflammatory cytokines that increases the inflammatory response in the brain, that could potentially be a bad thing. And it could also, then if that happens, it could dampen the beneficial effects of the treatment. So that even though, and some people have had hypothesized that that could contribute to the relatively mild cognitive effects you see, that you remove the amyloid, but instead you increase the inflammation, potentially. So we would like to study that in more detail and see how, what kind of response do we get specifically from microglial cells and from astrocytes when you do this treatment. And you can study that also separately in cell cultures, for example, where you culture a certain specific type of cell and then you give an antibody treatment together with A-beta and see what kind of response you get from it.
Yes.
So it's a good question, and there have been different types of antibodies have been studied with different degree of effector function, as it says, or as it's called, where some antibody types are less prone to trigger microglial responses. And to my knowledge, it hasn't been completely resolved which type of antibody is the best and what effect it has specifically. I think it's difficult to say from these clinical studies. You can never test, let's test 4 different antibodies in a clinical trial. You have to go for one, and then it comes with all its different features, and you test all of it together. But I think that the link with neuroinflammation and how treatment affects neuroinflammatory responses are things that are important to study.
Yes, I see. In case that, because to my understanding, you're saying that there are some people that are hypothesizing that it's leading to this cytokine release, and that might be responsible for, a relatively, to what the effect would be otherwise, small effect on cognitive function. This might be a curveball question for you, but if you were to give one of these antibodies and that would be a reason for a lessened cognitive benefit, couldn't you give like a strong anti-inflammatory that could reach the brain, like some form of cortisone co-treatment?
I think that, this is actually done for those patients that get severe side effects. So I read recently that a hospital in the United States that treat patients with Lecanemab have found out that they can actually control the side effects with steroid treatment. And that is a way to dampen the inflammatory response that gives rise to the ARIA.
Yes.
But I don't think that anyone has tested. You can imagine that you would have a combination treatment where one targets the immune system or the inflammation. But I don't think that that cortisone, for example, treatment has been tested or postulated as a treatment for for a co-administration with antibody treatment. I think that maybe it has to be a little bit more specific, so you find the mechanism that you want to target specifically, which you probably then have to study in separate preclinical studies, and then you do a clinical trial with the antibody and the combination.
Yeah.
And one reason that it's been difficult to do combination studies is because you need to assess each drug independently.
Sure.
So the Lecanemab or the anti-A-beta antibody needs to be shown first to have a treatment effect, and then you can combine it with other things.
Yes.
So now, in fact, there is a study running with Lecanemab and a tau antibody.
Okay.
As a combination therapy.
Yeah. Yeah.
So that would be exciting to see if that has an increased effect.
Yeah. So tau is also a target for treatment
yes.
for Alzheimer's?
Yes. Yeah. And I believe that tau is even more difficult than A-beta for treatment. Tau has a very defined physiological effect, and you need to avoid to treat the physiological tau, because otherwise you might destroy the neurons. So that's one of the important aspects. And then also the fact that tau is to a large degree intracellular.
Yes.
So
another step.
There's another step.
Yeah.
Or you need to target tau aggregates, for example, that are released in the extracellular matrix. But it adds to the complexity, I think.
Yeah.
So, but we'll see. Tau is like a decade after A-beta.
Okay. I was going to ask that actually.
Yeah.
So it's about a decade after. Yeah. So maybe in 10 years we'll see some approved.
Potentially. Let's hope so.
Let's hope so. Yeah.
Because I think in the end that, as you say, combining therapies will probably be necessary, and you can then. I mean, we often discuss Alzheimer's disease as a disease, but as I touched upon in the beginning, it can be very heterogeneous. So I think that if you can then stratify Alzheimer types and say that this type of Alzheimer's disease would benefit from this treatment and this treatment, whereas this type would benefit from this combination of treatment.
Yes.
Or you could say that, for example, it would be good to start with an antibody that clears the amyloid plaques, and then you can give a different medication to keep the brain healthy afterwards. So, yeah. So the future will tell, probably.
Yeah. So I was thinking we can move a bit onwards towards, or I guess back in time, relative to when the disease has already been initiated and started progressing. What can people do in general to reduce their risk of developing Alzheimer's before even getting the disease?
Yeah, I think that a general type of advice that you give for more or less all types of diseases, like cardiovascular disease, cancer, is to eat healthy, exercise, sleep, and so on. And I think, although there are studies suggesting that if you have a healthy lifestyle with exercise, good nutrition, and social interactions, you might delay the onset of dementia, for example. But I think it's unclear whether that really has an impact on the actual pathological process, or if it's more that in doing these things you are better at coping with this pathological process. So your brain can take a little more insult before you are affected by it.
Mm-hmm.
And this is a little bit a matter of debate, and obviously it never contradicts the advice to have a healthy lifestyle, but it potentially contradicts that you have the possibility to prevent the development of Alzheimer's disease. And I think because we don't know why this build-up of amyloid-beta, for example, why it starts. In some cases we do know when we have familial disease with a mutation in a protein, but this is very, very rare. So in the vast majority of cases we don't really know why it starts. And as also with the cardiovascular disease, you might have had a perfectly healthy life and done everything right, and you have no known genetic risk factor, and then you're affected anyway. So it's the same here, and I think it could be that you can sort of slow the progression, but it could also be that you're only improving your brain's ability to handle the problem forward.
Mm-hmm. Okay.
Which is also good, obviously. From a general health perspective. But I think it's important not to tell people that you can avoid it if you do the right thing. Because then you also say, if you get it, you have done the wrong things.
It's your fault.
It's your fault. And I don't think that's true.
I
because
agree.
it is a disease that you can get.
And that's not the point of this discussion.
No, obviously. Obviously. So I think definitely do all these healthy things. It will definitely be good for you, and it may delay your dementia, but it may not slow the course of it.
I see. So if I understand correctly, there are no disease-modifying lifestyle interventions.
And I would say that there are none that has been completely proven, but I'm also a little hesitant because this is not my main area of research, and there may be research that I'm not aware of.
Sure.
But at least if there was a miracle cure, we would all know about it. And we don't. But there are clinical trials, for example, of nutritional additions. For example, you eat certain things that should be, for example, neuroprotective or so. That may have some effect. But again, it's difficult to say whether it has an effect on the development of pathology or on how the brain can cope with it.
I see, because it's like a homeostasis between the progression and the coping by the brain with the stress of that progression. And
it's in a way similar with the existing Alzheimer medications. We have two that have been around for 20 years or so, or more. So one of them is acetylcholine esterase inhibitor, which improves the acetylcholine signaling in the brain.
Yeah.
But it works only as long as there are enough synapses to help, so to say.
Sure.
So it improves the symptoms, basically helping the brain to cope with neurodegeneration, but it doesn't change the course of the disease.
Right, so cognition is preserved only when the disease hasn't progressed so far that there are no synapses, no neurons left, basically.
Exactly.
Yeah, I see. We started this conversation with talking about hippocampus and the role of hippocampal neurodegeneration in early stages of the disease. I was thinking about this yesterday, actually, and to my understanding, cardiovascular exercise and also SSRIs can both promote hippocampal neurogenesis. Might this be a way of, like, just the mechanism in general, finding interventions that promote hippocampal neurogenesis? Might that also be a method for promoting the brain's ability to cope with the increased stress of the pathological processes in Alzheimer's?
I think this is an interesting view and an interesting question, and I must admit that it's a little bit beyond my research expertise, although I think in principle, as you say, if you can induce neurogenesis, it could be beneficial. But you might also ask or wonder whether neurogenesis can help in replacing connections between neurons in the brain that have died.
Yeah.
So, to my mind, it may be slightly different things that you have neurogenesis and that you have dysfunctioning and dying connections between neurons. But as I said, this is a bit outside my expertise. So there may be things here that I don't know, and I think that that is definitely something that is interesting to explore.
Yes. So an interesting topic of research to explore, and maybe there might even be some evidence already. But it might also not be as simple as just replacing the neurons that have died, because there are so many connections between the neurons, and those would have to be fine-tuned as well.
Yeah.
Yeah.
But there are examples, I mean, in Parkinson research, for example, where cell replacement therapies are ongoing and tested, where you have dopamine-producing cells that you introduce to patients, and that where you can actually, I believe, see some benefit. So it's not. I believe that it's more difficult with Alzheimer's disease, because in Parkinson's disease you have these dopamine-producing cells in a very specific area. And if you replace them or you add new ones, they might grow out and replace those that don't work. Whereas for Alzheimer's disease, it might be more difficult to restore the connections that are needed. But I would not dare to say that it's not possible or so.
Yeah. Interesting. I also wonder, do you use artificial intelligence at all in your research group?
I wish.
You wish?
Well, we use it, as most people use it, like when you write things and when you want to find information and things like that. But not yet as more of a specific research tool. Yeah. I think it will definitely be very useful. You can imagine, I mean, in our area of research, for example, for image, for finding small changes in imaging. So brain imaging, for example, if
Yeah.
you do image analysis and you want to see how does this differ from this one. And the way we now do it regularly is that we measure the signal in a certain volume of the brain and we compare it to another one. Whereas if you use artificial intelligence, I believe that you could find much more subtle changes or patterns that you cannot see by eye or measure with the crude measurements that we use now.
Yes.
So that is one area where I believe that AI will be important. But also probably in handling of big data sets.
Yeah.
There is a lot of research now where you do single cell transcriptomics, for example.
Yes.
So you look at the protein expression, basically, or the genetic transcription of the whole genome, for example, of different cells on a single cell level.
Yes.
So if you use that for a brain, for example, you get enormous amounts of data, and I believe that with AI that can help you to sort and find patterns in this big data.
Yes, yes. I agree. That seems to be a major use case for artificial intelligence to efficiently interpret big data sets. And this transition may be a bit far-fetched, but and to get big data sets, it is important to work not only locally in your own research group, right? It's good to collaborate with different pharma companies, for example. You've had experience both on the side of working in pharma companies and also in research groups at universities. From your experience, do you think that there are any levers that can be pulled as regards ways to improve how research is conducted, both separately and also in the collaboration, to facilitate effective drug development?
Yeah, I think that's hugely important because the academia and the pharma companies are fundamentally different in the way they work. So in the academic world, you identify a problem based on a research question, which can be anything, like a mechanism of action, or it can also be finding a drug to something. Whereas in a pharma company, you strive to develop something that can help usually a large population, but that can also bring in a benefit.
Yes.
So a revenue to the company.
Yeah.
And the company needs to have something they can sell.
Yeah.
But the companies also have the ability to do this drug development, which costs a lot of money and has to include a lot of people, which is much more difficult to do in an academic group. It's not impossible, but I do believe that the collaboration between academia and companies, if done in a good way, definitely helps the process. Because I think that the innovation is so much broader in the academic world, where people look at everything, anything out of interest.
Yes.
Because they have found something that leads to something else, and so on. And pharma companies can never work in that way. So pharma companies need academia to find the very innovational drug targets or processes to target with drugs, and academia needs the companies to develop the drugs.
Yeah.
And we actually have at Uppsala University, we have Uppsala University Innovation, which is a unit that helps researchers at the university to either start their own companies or to connect with existing companies and make deals and with patenting and so on, because it's very important to patent things at an early stage so that it can be protected during the development phase.
Yes.
And so on. So I think that it's not necessarily very easy, but I think it is necessary for drug development to have this collaboration between academia and industry. And I also think that there is an increasing infrastructure to do this, and also people to a higher degree are aware of this nowadays.
Yes, yes. I see the value of it too. Have you had any pain points in collaboration with either pharma companies when working at universities, or universities when working at pharma companies? Do you have any, like, I would like this to be improved it would be so much more effective, the drug development process in general, if we could just do it this way instead? Or do you think it's already a pretty optimized process?
It's probably not an optimized process. My experience from working at a pharma company is relatively limited to a very early point in my career when I was an industrial PhD. And since then I've worked mainly or exclusively at the university. I think that communication and openness is something between the parties.
Yeah.
That you can talk about things and that you can discuss things freely. You don't necessarily have to disclose things to other people, but to have like a good communication between the parties involved. That of course is very important. And then sometimes it's obvious that you have different objectives. Pharma companies want to patent, they want to make a product, and not necessarily to find out exactly how everything works. Whereas for the academic researcher, you might be less interested in waiting for a year to disclose your results. Maybe you want to go to a conference and say, we have found this, or you want to publish it immediately, and that there may be restrictions and you have to agree. And also when you have found something, you want to dig deeper.
Yeah.
And then you might find something that is not necessarily good.
Oh yeah.
And obviously everyone is interested in the end in finding drugs that are efficient and safe.
Yes.
But there may be some things that are perceived as potentially a problem in terms of efficacy or safety that is found by the academic research, and that maybe the company's marketing division are very afraid that this will harm their product. So this type of conflicts can arise, and I mean, it's natural because we have different goals.
Yeah, different incentives.
Has to be discussed and communicated around it, and you have to have mutual understandings about things without compromising integrity and quality of research.
Yes, yes. That sounds like the only way to do it, I think, to have this open communication and to on a case-by-case basis, balance openness and transparency and getting all the data out there. For example, with letting the companies not present necessarily all the data, or even to invest in, for example, finding mechanisms if they don't think that would be an economically reasonable decision for them. Yeah.
And then another aspect that comes with collaboration between academic world and pharma companies is financing of studies, which can, if we look at Denmark, for example, they now have a huge pharma company, Novo Nordisk, which is bigger than Denmark, basically. And they also give out a huge amount of research grants. They have a foundation which finances a lot of research, and that is not that's open. You apply for anything and you can get a grant. Maybe within a certain area of research. I mean, they are interested in metabolic diseases, so perhaps there's mostly focus around that.
Yes.
But that's also a way for the companies not only to give back to society, but also actually to bring in new potential research that they can use for further development of drugs.
Giving grants to researchers at the universities?
Yeah. So even though they don't control the research at all, I mean, they give money with no restrictions. Here, use it as you wish, as long as you follow your research plan.
Yeah.
And then there might pop up very interesting things in the research groups, and then they can perhaps make a deal and say, oh, we would be interested in developing your results into a drug.
Yeah.
So I think that that's also like a very smart way from the company to boost the academic research and at the same time potentially have benefit back.
Yes.
So that's something that I think could be perhaps even more done also in Sweden.
Yes. Sounds synergistic.
Yeah, I believe so.
Yeah. All right, so the final question for the conversation. Do you think that there will be a cure for Alzheimer's in your lifetime? Like a complete cure?
Well, I hope I will live long enough to see that. I will give you a researcher answer.
Yeah.
It's very difficult to say a complete cure, but I think it's reasonable to believe, provided I live long enough, that there may be treatment that halts the disease processes enough to keep you on a relatively stable cognitive level throughout the rest of your life. I believe that's reasonable. So I'm leaning towards a yes-ish answer there to your question.
Sounds optimistic.
Yeah. I think that the past 5 years have been very optimistic. So I think we should see it that way.
Yes. Right. Thank you, Dag Sehlin, for coming on the Exploring Health and Medicine podcast.
Thank you very much for having me. It's a pleasure.